can take, right? I can take photos. Puedes tomar.
Oh, yes. ¿Me escuchan? Ok. Voy a sacar eso para hablar más claro. I have a big pleasure to present today, to introduce you Professor Gyorgy Panyi from the University of Debrecen in Hungary. Gyorgy has been a professor and head of biophysics at the University of Debrecen Faculty of Medicine, Department of Biophysics and Cell Biology, Hungary. Since 2009, I guess you were there, right? Dr. Panyi has a graduate as a medical doctor in 91 and had his PhD in 96. And he spent 2 periods at Pennsylvania University, 2 periods of 2 years. Actually, part of his thesis was done there. And then he presented the thesis in Hungary. Our relation is very old. It's almost 30 years. Actually, our first paper was published in 98. So we started working before that. So this last century. But the collaboration has been very, very fruitful. I must say that we have about 20 papers published in international journals. We have 20 patents registered and approved of the work done in collaboration with him. And actually, many of the students that he worked together with us in the department got their PhD thanks to this collaboration. So for me, it's really a pleasure to announce that we have the opportunity to have him today here giving us a lecture. And I must also say that this is supported in part by the Hungarian embassy here in Mexico. This week, they are celebrating a joint effort of Mexico and Hungary. And for that reason, the embassy also helped us having Dr. Gyorgy Panyi here as a visitor. And I personally also have another reason to be very glad to have him here. If you notice my name, family name, and as a Postai, Posani Postai, that is a Hungarian name. One of my parents of my grandparents came from Hungary and to south of Brazil. And so there is a second reason for me to be happy to have Professor Panyi here to deliver his lecture. Please.
Thank you. So thank you very much for the very kind introduction, and I'm really honored to be here. And thank you very much for the nice words from Professor Posani. I mean, indeed, we have been working together for a long time on scorpion toxins, and I'm going to be talking about scorpion toxins as well. And I hope that I would be convincing you very successfully that this is basically the future of medicine, that we are going to be making scorpion toxins to treat various diseases, including the inflammatory diseases and also some neurological diseases. Okay, so can you hear me well? Is it okay? Good, excellent. So the title of my talk is Scorpion Toxins Targeting Kv1.3 with the therapeutic implications. So I'm going to be talking mostly about our research together with Professor Posani, and also giving you some motivation to that by going into some of the diseases that can be targeted. So I will talk about 3 things in this talk. It's going to be T-cell ion channels, which is going to be the introduction. Then there is going to be something about the diseases that we are targeting, and there will be the most of the talk is going to be about the how we are developing these peptides. Now, I would like to start my introduction with a very small, a few figures about the ion channels that we are going to be discussing today. Most importantly, I would like to introduce you to the Kv1.3 ion channel, which Kv1.3 ion channel is expressed in homotetrameric form in lymphocytes, also in some of the immune cells, and also in heterotetrameric forms in neural tissues, and this is what I'm going to be also introducing you to. The other ion channel that we have been interested in, mostly to get rid of the target as a potential target, is KCA3.1, which is the calcium-activated potassium channel of lymphocytes. And these two ion channels are going to be the key players for regulating T-cell activation in many of the diseases. This is just a little bit of biophysics to introduce you what the major differences are between these two ion channels. One of them, the voltage-gated potassium channel, is voltage-gated, which means that at different membrane potentials, you have different magnitudes and kinetics of the current that we are recording using the patch clamp technique in human T lymphocytes. The other ion channel is calcium-gated, not voltage-gated. So it has a linear current voltage relationship and also very high sensitivity to calcium concentration within the cell. This calcium sensitivity is conferred to the channel by the presence of calmodulin, which is permanently attached to the C-terminus of the ion channel. The interesting part is that these two ion channels play a key role in T-cell activation. And just to make it very general and very brief, upon presentation of the antigen to the T cells, this antigen presentation is going to be followed by a calcium signal, which is delivered by the ORA channels, which I'm not going to talk about today because we are not dealing with the ORA channels. But as a consequence of this calcium signal, there's going to be T cell activation and T cell division and clonal proliferation of the cells, which are specific for a given antigen. Now, how do potassium channels come into this picture? Potassium channels are regulating the membrane potential of these cells. The membrane potential is clamped around minus 50 millivolt as a consequence of these two ion channels. So, what happens when we inhibit potassium channels? When we inhibit potassium channels, then the membrane potential will change, and the change in the membrane potential will influence the calcium signal, which will be inhibiting. If you inhibit the calcium signal, that will also inhibit the T-cell proliferation and activation. So this small video is going to be showing you a very nice record of T-cell activation in live. So this guy is going to be the T cell. This guy is going to be the antigen-presenting antigen-presenting cell, and follow the color of the T cell as it's interacting with the antigen-presenting cell. The color is proportional to the calcium concentration. You can see very clearly that upon T cell activation, there is a huge calcium signal which can be inhibited by potassium channel blockers. So. Now, what can we use these potassium channel inhibitors for? If we can inhibit the potassium channels, and then we can potentially target immune and autoimmune diseases, because during the autoimmunity, self-antigens are going to be recognized, and destruction of the self-tissue, myelin, or the cartilage in the joints is going to be destroyed. And we are targeting this to use the ion channels inhibitors as potential immunosuppressors. Now, the problem with immunosuppression that if it's general, then it's going to be affecting other immune reactions. Especially if you have a foreign antigens, the bacterial infection or viral infection, you're going to be suffering also if you have general immunosuppression. In the following slide, I would like to show you how potassium channel inhibitors are better than a general immunosuppressor, how they are better than cortisol or any of the general immunosuppressors. This is going to be shown on the next slide, where I will show you what happens when you have a chronic antigen stimulation, usually present in autoimmune diseases. So the naive T cells, which are going to be repeatedly stimulated by an antigen, they are going to be differentiating towards central memory T cells, indicated as TCM, and then finally they are going to be differentiating into the effector memory T cells. Now, these effector memory T cells has a very unique ion channel repertoire. They have Kv1.3 and KCa3.1 potassium channels, but Kv1.3 at a much larger quantity. And once you are activating these effector memory T cells during autoimmune reaction, then the potassium channel repertoire is going to be selectively changed, where effector memory T cells are going to be expressing a very large amount of Kv1.3. So these cells, the effector memory T cells, are characterized by Kv1.3 high, IKCA1 low phenotype. And these phenotypes is also found in human tissues. So if you look into this very old image, which was the first demonstration of this, the presence of these cells in post-mortem multiple sclerosis brain slices, these perivascular infiltrates are going to be very high Kv1.3, and this one is also very high in Kv1.3 from the infiltration of the T cells. Another very common autoimmune disease is rheumatoid arthritis. And then if you take the liquid from the joint of rheumatoid arthritis people, then you can also get T cells, and you can study the T cells from patients. And you can see that rheumatoid arthritis patients will have a very, very large Kv1.3 expression as compared to other diseases of the joints or any other T cells from the same patient. So what the most important of this slide is that you will have a unique ion channel phenotype with around 1,500, 2,000 Kv1.3 channels per cell. And then we would like to target these specifically. If we can target those, we can inhibit selectively the effector memory T cells, then we can get immunosuppression. So now to target Kv1.3 selectively, we need drugs, and these drugs are with the long-term collaboration with Professor Panyi, we are targeting them with scorpion toxins, and this is taken roughly 20 years ago in Professor Panyi's lab. I was younger, also probably 20 kilos lighter, and then I have this scorpion on my hand, which was just anesthetized and was ready for milking, so that we could get the scorpion toxins out of them. So our idea is that we are going to be placing the scorpion into the infusion vial, and then we are going to be taking the medicine out of it, and then it's like the snake wine the Vietnamese are making. They put the whole snake into it. We don't put the whole scorpion. We are going to be going to separate them, and then we are going to be seeing them as peptides. So the take-home message of this slide is that Kv1.3 inhibitors could be selectively immunosuppressive drugs, which are targeting the effector memory T cells, and most importantly, the Kv1.3 channel in the effector memory T cells. As you can very clearly see, we would like to have a high selectivity. We do not want these drugs to inhibit KCa3.1, because if you inhibit the calcium activated one, then you are going to be also inhibiting the normal immune responses. So this is something that you would like to avoid. And now we are going to go to the second point, which is going to be the diseases. As I already mentioned one, which is multiple sclerosis. In multiple sclerosis, I have shown you the T cells which are infiltrating the brain. And then there are a lot of in vitro and also in vivo mice studies that are reporting on the beneficial effects of Kv1.3 inhibitors on multiple sclerosis. Something which has already been shown, but not in details. This is the arthritis model. An arthritis model can be also inhibited. In the arthritis model, the consequences, this is a diseased model. This is a diseased animal. And this diseased animal is characterized by deformations of the joints because of the autoimmune disease. And this can be very clearly treated by selective inhibitor of Kv1.3. So you can see very clearly that the treated animals do not have these deformed joints, and their affected joints is going to be very much smaller in the Kv1.3 inhibitor treated population as compared to the control. A little bit more on these immune-mediated inflammatory diseases. There are several of them: rheumatoid arthritis, juvenile arthritis, interestingly, uveitis, psoriasis, IBD. These have a common disease mechanism. This is going to be the activated T cells, which are going to be Kv1.3 high, and theoretically, you can target all of these diseases by Kv1.3 inhibitors. So these are the various forms of rheumatoid arthritis, and the skin diseases, and the spondyloarthrosis, and the gut diseases, and IBD, for example, most importantly, IBD. And the common mechanism is that there is T cell activation. There are certain T cell phenotypes which are going to be activated with Kv1.3 high, so that they are ideal target. Obviously, we have to avoid macrophage targeting, which is going to be later discussed in the talk. Also, less frequently discussed is the psoriatic disease. Psoriasis is also characterized by infiltration of the T cells, having Kv1.3 high phenotype. So these are also Kv1.3 high cells. So this is a skin section, and this is Kv1.3, and this is CD3. So you see that the CD3 positive Kv1.3 high T cells are going to be accumulated into the diseased skin. And also, if you are isolating T cells from this disease model, then you will see that there are very, very large currents, potassium currents, very much larger than in other forms of either in control cells or in other diseases. So the most important is that you are again seeing a Kv1.3 high phenotype in psoriatic diseases. And if you are using an Omniment, which contains a Kv1.3 inhibitor, as it was discussed in this paper below. So POP1 is a small organic compound, which is also a very high affinity Kv1.3 inhibitor. You can basically treat the disease, and you can get back to a normal tissue as compared to the very highly CD4 T cell infiltrated disease model. So on the left-hand side is the disease, and then on the right-hand side is after the treatment with Kv1.3 inhibitors. So the major message from this part of the talk is that if you can target Kv1.3 with high affinity and high selectivity inhibitors, then you may be able to influence several diseases, including the rheumatoid arthritis, type 1 diabetes, which is also an autoimmune disease and related to the T cells of God65 positive T cells, and also psoriatic disease and uveitis, and most importantly, or probably IBD as well, in inflammatory bowel disease. So having that discussed, now it is time to go into the particular details and seeing what type of ion channel inhibitors can we develop, and what are the sources, which was already discussed, that we have a source is the scorpions. And then I would like to go forward the mechanism of those scorpion toxins. Now, if this is the ion channel that we are targeting, and this is the toxin, which is a peptide, that's what we are dealing with when we are isolating from scorpions. These toxins are going to be plugging the pore of the potassium channels, or plugging the pore of the channels in general. So these are the pore blockers. This is potassium flux, and potassium flux can be terminated. Statistically speaking, if we have a lot of ion channels and you apply the toxin at half inhibitory concentration, you're recording the ionic current from a T cell. I just would like to mention that we are doing patch clamping and we are recording ionic currents from T cells and also many different kinds of ion channel currents from heterologous expression systems. And what you can see is that the whole cell current is going to be reduced. Now, if you move from this very schematic view to a more detailed structure of these toxins, which was usually determined by either NMR structure, mostly by NMR structure determination, and we go to the structure of the potassium channel, which was done by X-ray crystallography and also cryo-EM, then we can recognize this very nice complementary surface between the toxin and the ion channel. Thereby, the toxin can plug the pore like a cork in the bottle. And the toxins have a few very important characteristics in general. They have an alpha helix and several beta strands, 2 or 3 beta strands, depending on what kind of toxin we are talking about. And the structure of the toxin is stabilized by disulfide bridges. And they also are characterized by so-called essential diode, which is a lysine, which is protruding into the pore. And there is an aromatic residue very close to this lysine within a few angstroms of the alpha carbon of lysine. So during the block, this lysine is going to be protruding into the pore, displacing potassium channel from potassium ions from the selectivity filter, mostly in the first selectivity filter binding site, potassium binding site, roughly from here, and then this will disable the potassium channel and will stop the conduction. Very good. So what is the problem with finding a specific target? So the problem is that sometimes you have to get, you would like to get a very highly selective potassium channel inhibitor, which will theoretically only inhibit Kv1.3. What is the difficulty? The difficulty is that if you are looking at the Kv1 family of ion channels, this is where the toxin receptor sites, starting from the turret and the selectivity filter. So this region is going to be the toxin receptor. They are very similar in the toxin receptor site. So the red indicates the differences, black indicates the homologous parts. Therefore, looking at the sequence, you can imagine that it's extremely difficult to find selective inhibitors. Now, this is the workflow that how we are going to be targeting this problem. And I'm going to be referring back to this slide many, many times during my talk, because on the left-hand side, I have the sources that we are working with. This is the way how we are going to be making the peptides. Either we are purifying them, either we take the mRNA and then we do the transcriptome analysis and then we get, or we have known peptides and then we do recombinant or synthetic production of the peptides. Once we have the peptides, we use patch clamp electrophysiology to characterize them, and then we do the biological assays in order to find how these peptides are going to be influencing the function of the T cells or on the level of the animals. So let's go to the first one. The first example is going to be the one that Professor Posani has already referred to. So this is our best compound that we have found so far. We were trying to identify natural sources of peptides from scorpions. They did the purification here and also sequence determination and the structure biology in Mexico. We did the ion channel assays and also the biological assays were done here by Professor Ivan Rosenstein. And I wish her the best for quick recovery. This is the guy that we were studying is Vaejovis mexicanus, smithi. And this is the work by Professor Posani, and they have milked the scorpion. They did the HPLC purification, and they identified many, many, many different peaks. One of the peaks, I mean, we also tested many of these peaks, but one of the peaks was very promising. They determined this primary sequence of the amino acid sequence of the peptide. It consists of 36 amino acids, structurally stabilized by 4 disulfide bridges. And it looked very similar to the other potassium channel inhibitors because it had the disulfide bridges, potentially alpha helix and beta strands, and also the essential diet for T cell, for the inhibition of potassium channels. That's why we went for electrophysiology. And we were very much surprised. The big surprise came that at 1 nanomolar concentration, which is this is how we start testing the peptides, 100 nanomolar or 10 nanomolar or 1 nanomolar. And in this particular case, 1 nanomolar of VM24 inhibited the potassium current fully in Kv1 in T cells. This is the Kv1.3 current that we have been measuring. Another very interesting, if we went, I mean, this was a full block, so you cannot really get any detailed information. So we had to get very, very low concentrations. And interestingly, at 3 picomolar concentration, this peptide is already inhibiting Kv1.3. And then you see that the wash in kinetics, this is the peak currents as a function of time. The peak currents are going to be gradually decreasing. And at one point, our colleagues got tired, and then they switched back to the normal extracellular solution, and they tried to wash out the peptide. So basically, the peptide it's very difficult for the peptide to dissociate from the ion channel. It has such a tight binding to the pore that the toxin doesn't want to get off. It was extremely difficult, therefore, to get a dose response curve, because for a dose response curve, you need reversible blocker and to get equilibrium at each concentration. So we did a cumulative dose response curve, and then we tried to do our best. Most likely, the toxin is more potent than the one that we are reporting here. This is how far we could wait for each current block and development of the quasi-equilibrium. So it's not a full equilibrium, it's close to the equilibrium. But from this dose response curve, we could get a KD around 3 picomolar, which was very, very unusual at that time. And then this is still the highest affinity, one of the highest affinity Kv1.3 inhibitors, and the highest natural one, the ones that are not modified, because there are others in the market which had to be modified in order to be that high affinity, and also to get that very high selectivity. So if you look at the selectivity profile of this drug, and now we are testing this drug at the VM24, we are testing at 10 nanomolar. 10 nanomolar is 3,300 times higher than the KD. So around 3,000-fold the KD concentration. It didn't inhibit a large battery of ion channels. It inhibited Kv1.1, 1.2, and Kca 3.1, around 50% if you do 3,000 times or 3,300 times the KD value for Kv1.3, which means that this is a very selective peptide. If you go to 1 nanomolar concentration, then you barely see any block of the other ion channels. It's only Kv1.2, which was a little bit inhibited. So among the natural peptides, VM24 is the best, which far the best in the market at this moment, with its very, very high affinity and very, very high selectivity. And then with the help of Professor Posani's colleagues, Ricardo, we have also made some docking of the peptide into the Kv1.3 pore. At that time, we didn't have the Kv1.3 structure, so we had to use a homology modeling of Kv1.3, and then we docked the peptide into this pore of the channel, and we try to identify those interactions which seem to be unique for Kv1.3 and VM24 interaction. And if you are looking at, again, to the sequence of the peptide, we found some of those specific interactions between corresponding residues on the Kv1.3. I'm not going to go into the details because these interaction points are just predictions based on the docking simulation, and we do really need to get a little bit more data from cryo-EM for directly observing the interaction of the channel and the toxin. Now, this work was so far the electrophysiology and the structure determination and the affinity determination were done mostly by Zoltán Varga from my laboratory and also Ferenc Pap. And I'm very grateful for Georgina Grola, who is also here in the audience, who did mostly the biochemistry and also the synthetic generation of this peptide for the structure determination. And also it was part of the big efforts to docking. And with Ivan Rosenstein here, there we did a lot of functional studies. And these functional studies showed that VM24 at picomolar concentrations, very similar concentrations to the ones that are used for Kv1.3 inhibition, they also inhibited T cell proliferation in blue, the current in red, and also CD25 expression in green, which is an indicator of T cell activation. So in other words, we have a drug which is in the same concentration range as the Kv1.3 inhibition. It inhibits T cell activation markers and also inhibits calcium signaling. So this is the induction of calcium signaling in T cells, and VM24 completely inhibited the second phase of the calcium signal, which is expected from a drug that interferes with T cell activation. So there is a very important message here that in vitro we can get the T cell activation inhibited. And we were also very lucky to show that in vivo, the drug also works. So if we go to delayed type hypersensitivity reaction, which we also did here, and we also had a colleague in Kristin Beaton to help us with the experiments. So if you do a DTH reaction, which is effector memory T cell mediated inflammation of the ears, because you add the antigen to the ears, what you see is you will have the ear swollen, and you can measure the swelling of the ear, and that could be a very important, very interesting readout for the disease progression. And also, if you add the toxin VM24 to the animals, you can see how the ear thickness is going to be reduced. So this is what we were looking for. And this is what we have shown. This is, as a matter of fact, I think this is my fingers on this animal, and we did this experiment here with Luriva, and also I think Georgina was also there. But this was a very risky experiment because the rats didn't like their ear to be measured, especially when it was inflamed. So it was very painful. So we had to be very careful with that. So the interesting message is, if you have untreated animals, you see very clearly the infiltration of the ear. You can see the vascularization, the red and swollen ears. If you go to a treated, where we injected the rats with 10 microgram of VM24, then you can see that the ears are transparent, no vascularization, no infiltration from the T cells, and we can also see that difference numerically. So we can do the measurement, and we can, using this caliper, we can determine precisely how much the inflammation has been reduced by the application of the toxin. So the take-home message from this part of the talk is that we can find natural peptides which are very highly selective, very high affinity for Kv1.3, which do have the which do inhibit T cell activation in vivo as well as in vitro, and then we can do quantification of these effects. Now, the next part of the talk is going to be what happens if you find a peptide and then it's not selective. How can we make it more selective for Kv1.3? And this is also the story was originated from this laboratory with Professor Possani. We found a peptide which was non-selective for Kv1.3. So this peptide is called Anuroctoxin, and we published this paper in 2005 with Professor Possani's lab. And then what you see on this graph is that this guy is going to be inhibiting Kv1.3 and Kv1.2 almost the same extent. So almost the same affinity for the two ion channels. Now this one obviously cannot be injected, because if it's Kv1.2 is expressed in many places in the body, so you would like to get a therapeutically applicable drug. Thereby, we had to figure out how to increase the affinity. So now we did a lot of this sequence analysis with Gerardo Corzo and with Professor Possani in previous papers, and we figured out that there is some pattern for the Kv1.3 specific inhibitors, namely that there is this threonine and there is alanine in position 17. So we decided, okay, let's try to tailor the selectivity of Anuroctoxin towards Kv1.3 by making mutations. So what happens if we make this N17A F32T substitution of the toxin? And this was a very simple study. We synthesized the toxin in the control and in the mutant version. We decided one by one or in combination, and we did the dose response curves. So what you see on the dose response curves is, as compared to the wild type, N17A reduced the affinity, F32T also, and then when you have the combination of these two, those two are going to be running almost on the same dose response curve as for Kv1.3. Therefore, you will have a very high affinity Kv1.3 inhibitor with the double mutant. So what happens to the selectivity? Because we anticipated that if we make these mutations, we will convert the toxin to Kv1.3 selective at 1 nanomolar concentration. The double mutant, double substituted peptide blocks around 50% of the current. If you go to 100 nanomolar and Kv1.2, there is no block. So basically, the one that we had previously, similar affinity, we made the double substitution based on analysis of the sequences, and this double substitution resulted in a very, very highly Kv1.3 selective, and this Kv1.3 selectivity was retained. So if you go to other ion channels like Kv1.1 or KCA3.1 or Kv1.2, Kv1.3, if you look at this graph, the only difference is between the wild type and the and the double substituted one is that it lost the affinity for Kv1.2, but all the other selectivity issues remain. So in other words, if you are looking at the non-selective peptide, a non-selective peptide can be converted into a Kv1.3 selective peptide if you do double substitutions or mutations in the toxin sequence. Now, we were very much interested in this phenomena. So how can we make this very simple mutations? Well, how do they affect the structure of the Anuroctoxin, and can we get any information from the structure? And this is the wild type, and this is the mutant. Double substituted. If you look very carefully, there is one thing which is very apparent from the structure. So the double substituted peptide becomes much more rigid. So the wild type has a very large structural flexibility, which is completely reduced with the double substitution. And these double substituted peptides, we can do the residue-wise root mean square deviations of the atomic coordinates, and you can very clearly see that the Y-type toxin has a very large flexibility as compared to the mutant one, which is in white bars. So we ended up with the conclusion, and we also supported with molecular dynamic simulations, that this reduction in the flexibility can be one way to increase selectivity for a given ion channel. You can imagine that if you have many, many different conformations, you can fit into many, many pores. If you are very rigid, even more rigid than the normal peptides, then you can probably fit into one pore only. And this is now accepted in the literature as a novel mechanism for increasing selectivity. This is reducing conformational flexibility, which can lead to the increase in the selectivity. So the take-home message from the second story is that you can make a conversion from a non-selective to a selective peptide, and then there is a novel mechanism which can contribute to this, which is the reduction of the conformational flexibility. So now we already did an increase in the selectivity by doing mutations, but there are a lot of peptides in scorpions. I mean, many of you are working with scorpions, so you know that there is a limitation to what you can find with HPLC. So you cannot get the miniature fractions. But now there is a new technology, transcriptome analysis of the venom glands, and fortunately, Professor Posani's lab is very good at that. So they were helping us, and then they were doing a transcriptome analysis of the scorpion venoms, and then the transcriptome analysis resulted in some new peptides. And these new peptides and this transcriptome analysis was published in 2015 with several potential potassium channel inhibitors. And one of them was VMKTX1, let's call it VMKTX1, which is very similar to the well-known VM24. I already spent several minutes with VM24, describing how good it is. And now we found they found, to be precise, with the transcriptome analysis, a variant of this toxin with a single amino acid substitution. But you see, this is a positive, this is a negatively charged amino acid, so it might have a very large effect on the affinity or the selectivity or both. So they made it synthetically, and they sent it to us, and then we did the electrophysiology recordings, and this is what a lot of stuff was done by Agota Choti, a PhD student of mine, and this stuff was published in Biochemical Pharmacology this year, this summer. So what you see over here is that if you start increasing concentrations of the synthetic VMKTX, then there's going to be a gradual reduction of the potassium current. This is what you anticipate from a potassium channel inhibitor. Unfortunately, a classical, I like to show this experiment because it's so nice. This is how good electrophysiology should be done. You have the drug, you add, there's the inhibition, loss of the peak current, and then you wash out, and you increase the concentration of the drug. You have many perfusion lines simultaneously in the patch clamp apparatus, and then you can see very clearly the reduction of the peak currents, and then wash out, again reduction of the peak currents, wash out, reduction of the peak currents. If you go to very, very high concentration, then you will lose all the current in one step, and then you can wash back fully if you have a fully reversible blocker. I mean, this guy unfortunately lost a very large part of its affinity for Kv1.3. So it's the KD is only roughly 0.8 nanomolar or 770 picomolar as compared to the 3 picomolar affinity for VM24. So we were not very much impressed. It was not very good result. However, when we went to the selectivity, And we wanted to, you know, when you went for VM24, 3,300 times the KD, you still had 3 ion channels was off-target inhibiting. Kv1.1, 1.2, and KCA3.1 were still inhibited by the drug, although at very high concentration. So these were the problematic issues. I mean, not very large problem, but still there could be some consequences of that. So we went for selectivity. Let's see if you lost a lot in the affinity, did you gain anything in the selectivity? And this was to our greatest surprise that if we go to 100 times the KD, or you go equivalently to around 3,000 times the KD value, you get eliminated 2 ion channels from the bank. So VM24 was inhibiting 1.1, 1.3, 1.2, and 3.1. SVM-KTX lost the affinity for 2 of them. The only bad guy is Kv1.2, which is very minimally influenced by this drug. So what we lost is affinity, but what we gained is increase in the selectivity. And interestingly, if you we went into the biophysical characterization of this block, and we figured out that most of the loss of the affinity comes from the increase in a washout. So the toxin washes in very quickly, but also washes out very quickly, which means that the negative charge we introduced will make unfavorable interactions with the toxin pore, with the channel pore. So the toxin can bind, but it's going to be kicked off much quicker. That's the explanation for the reduction in the affinity. And for the selectivity, we also did structure determination, and also we did modeling of the structure of the peptide. And the modeling of the structure of the peptide gave us, again, very similar finding that we found for Anuroctoxin. If you decrease the flexibility of the whole structure. Now this is VM24 simulation, and this is SVM-KTX. So if you are decreasing the flexibility of the conformational flexibility of the peptide, in other words, you make it more compact, it will most likely bind to a single type of ion channel. So again, decreasing the flexibility will explain the increase in the selectivity. And in this particular case, the reason for having this very much reduced flexibility is because there are hydrogen bonds that are generated between the newly introduced amino acids, so the negatively charged amino acid, and several other amino acids, as you can see it also on this illustration. So the N-terminus of the peptide was very much reduced in the flexibility, which gave us increasing in the selectivity, very similar to the Anuroctoxin that I have already shown you. So there are 2 examples for the same principle. If you have decreased flexibility, you have increase in the selectivity. Now, the next is going to be something which is very new for us. We have started it not a long time ago. It's only 1.5 years ago, and we have a little bit of results of that. And this is what I would like to share with you. And this is something that we are making a known peptide, which is going to be VM24, because this is our favorite. Okay, and then we are going to make it recombinantly and conjugate it with peptides, which will allow the penetration through the blood-brain barrier, and therefore we can also target neurological diseases. So what kind of diseases? Because I already told you the autoimmune ones, but I also told you that VM24, by targeting Kv1.3, is also a very attractive target in neurodegenerative disorders. And most importantly, the reason why we can use Kv1.3 as a very good target is because it has 2 actions. It will reduce inflammation, and it will also promote progenitor cell proliferation. So what is the common characteristic of these neurodegenerative disorders? There is a neuroinflammation, which is caused by microglia, and there is an impaired neurogenesis, which is the neurogenic progenitor cells are not going to be dividing, and they are not going to be substituting the dead cells. So if you want to target both things at the same time, Kv1.3 would be a very good tool, because as you see on this figure very clearly, the activated microglia, this pink colored dot is the Kv1.3 ion channel in the membrane. This activated microglia is going to be upregulating Kv1.3 and is going to be secreting the inflammatory cytokines. So if you can block Kv1.3 here, then you probably can also block pro-inflammatory cytokine production. On the other side of the coin is that, interestingly, the neurogenic progenitor cells, which were identified from disease models of neurodegenerative disorders, these NPCs, let me call it NPC because otherwise it's a very long name, these NPCs are very Kv1.3 high as compared to the healthy NPCs. So if you block Kv1.3, you can target both microglia and also you can target the NPCs, therefore reducing inflammation and increasing the replenishment of these neurons. And those simultaneous targeting can be done by Kv1.3 inhibition. And then I'm sorry for this very bad image. I should have checked it before. So there are two papers which are already promoting this idea in the literature. There are several more coming out, hopefully not quite quickly. And then the common mechanism that we are going to be targeting is that there is a blockade of Kv1.3, which will target neuroinflammation and also NPC proliferation. Therefore, you can, if you able to get the Kv1.3 inhibitors into the central nervous system, then you will treat or cure the neurodegenerative disorder. And the problem is that VM24 is very highly charged, small peptide. It's not going to go through the blood-brain barrier. These peptides are basically very difficult to get into the biological systems. Usually, when we do treat the mice, or not the mice, the rats, then we had to do intraperitoneal injections because they have very bad absorption through the guts, and also you can only give them parenteral manner. So in order to provide VM24 to go through the blood-brain barrier, which is an endothelial layer plus glia cells together, in order to promote the permeation, there are two known methods. One is that you are conjugating VM24 with a cell-penetrating peptide, or CPPs, that the conjugates will go through the endothelial cells. The other is that you are going to be conjugating them with the BBB shuttle peptide, which peptide has a receptor, and it's going to be a receptor-mediated transcytosis or absorptive transcytosis, or the receptor-mediated transcytosis. So either way, either using a CPP or you use a BBB shuttle peptide, you can get the peptides theoretically through the blood-brain barrier, and then you can inhibit Kv1.3 and do the NPC and also targeting the microglia. Now, the problem with that idea is that theoretically it's very easy to, so as we say, it's easier said than done, because the problem is that once you start conjugating the peptide with the carriers, either with the CPP, either the BBB shuttle peptide, you may end up losing the activity. So what you need to do is you have to first engineer the peptides, and then after you engineer the peptides, you have to show what the biological effect is. So we engineered these peptides by putting six histidine tags, a protease cleavage site, a BBB or shuttle or CPP, cell penetration peptide, a linker, and VM24 into a sequence. And then we conjugated with three guys: angiopep, is receptor-mediated. MTFP is also receptor-mediated, and TOT is an 11 amino acid base, absorptive. It's a CPP, the cell-penetrating peptide. When you make the conjugates, we tested them using electrophysiology if they work. Angiopep didn't work. Okay, so we conjugated with VM24. VM24 lost completely its affinity for Kv1.3. However, we were very lucky with the other two. And look, we also get even after conjugation and making a large modification on the N-terminus, you reduce tenfold the affinity from 3 picomolar, you go to 40 or 50 picomolar, but it's still extremely high affinity. It's still much better than anything that we knew before. So we are very, how to say, optimistic about these peptides. So we showed already that we can conjugate them. We can put either a CPP peptide or a BBB peptide with VM24. And once you conjugate them, you can maintain with these two guys, you can maintain the Kv1.3 affinity. What we are doing right now is we want to see if we can maintain selectivity also, and the most important would be to see how these peptides are going to go through the blood-brain barrier. This is work in progress. This is but I'm very happy that I can tell you here that we got a lot of very interesting and very promising data regarding the conjugation of VM24. And VM24 is very good in that sense because its affinity is 3 picomolar. So even if you lose tenfold, the affinity is still sub-nanomolar, way far than a nanomolar affinity. So the question is also, which is very interesting, so what are we going to be targeting once the peptides go through? Because don't forget that once the Kv1.3 and the peptide conjugate is going to go through the blood-brain barrier, it's going to be in the parenchyma and it's going to be in inhibiting ion channels. Theoretically, or in our hands, we are hoping that this is going to be inhibiting Kv1.3. But the problem is that in the central nervous system, Kv1.3 subunits are also expressed with other subunits of the Kv1 family in the neurons. So Kv1.3 is homotetrameric only in the T cells or the immune cells, but they can make heterotetramers. For example, in microglia, they can make heterotetramers of Kv1.3 and 1.5. And then in the neurons, there are Kv1.1, 1.2, and 1.3, which can also make heteromers. So the question is, what ion channels are these peptides going to be targeting? So can they inhibit neuronal activity? Because if they will, then we are in trouble. So there is a need to test this, and now this is what we haven't discussed with Professor Fossani yet, but now we are going to be going into the biophysical and pharmacological characterization of those ion channels which are made of heterotetramers under controlled conditions. So we are going to make, and we have already produced Y-type. This is the Y-type homotetrameric Kv1.3. This is Y-type in a form of dimers. So you are conjugating you make a concatemer of 2 Kv1.3, 2 Kv1.3. So basically you have a tetrameric Kv1.3 out of dimers. And then we are going to be making the heterodimers. The heterodimers are going to be such that we will have Kv1.3 in a dimer with 1.1, 1.2, 1.3, 1.4, 1.5, and see how our most important peptides are going to be recognizing those ion channels. this is very interesting. We have just started it. We have only one compound that we were asked by Ray Norton to test on this system because he was very much interested in that. Ray Norton is in Australia, and probably you know Ray Norton also very well. And he asked us to test on this model system already one peptide. So these are the first experiments that we did on the concatamers. And then we chose Kv1.3 and Kv1.5 as the targets because the Kv1.3 is has a very high affinity for this drug, but Kv1.5 is not inhibited by this HSTX variant. Let's call it HSTX variant. So Kv1.3 and 1.5 are very different because 1.3 is toxin sensitive, 1.5 is toxin resistant completely. You see the red and the black traces are overlapping, so there is no inhibition of the Kv1.5 current. The other issue is that they are very different in biophysically because Kv1.3 is inactivating with the 200 millisecond time constant, whereas the Kv1.5 is basically not inactivating. It's going to be inactivating with seconds of time constant. So when you make the heterotetramers, you would be able to see very, very clearly the difference. This is Kv1.3 homotetrameric, and this is 1.3, 1.5 heterotetrameric. So if you look very carefully, the inactivation kinetics of the heterotetrameric is quite different. Note the time scale. This is 500 milliseconds. This is 250 milliseconds. So if you take the inactivation kinetics of the heterotetrameric current, we are going to be seeing that it's around threefold slower inactivating the heterotetramer as compared to the homotetramer. So we can see very clearly that it is a heterotetrameric construct which is expressed. Now the question is, will it affect toxin binding? It should, because this is toxin resistant, this is toxin sensitive. The question is, what is the extent of the reduction of the toxin affinity if you compare this guy to that guy? So we had to do the pharmacology on the homotetrameric Kv1.3 made out of dimers and the Kv1.3, Kv1.5 dimers. What you see very clearly here is that the dimer, 1.3 dimer, maintained its very high affinity for HSTX as expected, because it should not change the toxin affinity, the dimer conjugation. But if you do 1.3, 1.5, it's going to be losing the affinity. Not that much, although it's only 18-fold reduction in the affinity, which means that even if you are combining a completely toxin resistant with a toxin sensitive one, only 18-fold is the reduction of the affinity, which means that this drug might also inhibit the ion channels which are formed with Kv1.3 and Kv1.5. So we have to do a very systematic study, and this is the end of my talk because I think it's already 1 hour. So that we need to, in order to target the central nervous system with these drugs that are penetrating through the blood-brain barrier, we have to do a very careful analysis of the homo and heterotetrameric targets and to eliminate the heterotetrameric targets, because those will inhibit the neurons, but those will also inhibit cells that we do not want to inhibit. Okay, so this is something which is a very important message from this. I do not have time for the last section, which was another peptide that we identified with Professor Possani, and I'm very grateful that it was also published in 2022 this year with my PhD student Umer Nasser, which was a novel peptide from a scorpion, Centroideus margaritatus. lotus, and which has a very unique structure, but unfortunately I do not really have time to discuss that. So, in summary, we have the following summary statements. VM24 is the most potent and selective Kv1.3 natural peptide. Okay? SVM-KTX is lower affinity, but higher affinity than VM24. We did change the selectivity of Anuroctoxin and tailored it for Kv1.3 during with a double substitution. We came to the conclusion that one way of increasing selectivity is that you are reducing the conformational flexibility of these peptides. And I also showed that VM24 can be successfully conjugated to BBB shuttle and BBB shuttle peptides. And I also called the attention that heterotetramers have significantly lower, but still only 18-fold lower affinity than the wild type to the wild type homotetrameric Kv1.3. Therefore, we have to go a very intensive study. I would like to thank my colleagues who have participated in this study. So Zoltán Varga is my long-term collaborator, and Ferenc, they were both here in Mexico, with Adam also being here in Mexico. This guy is Mexican, Chuy, you probably know him from the institute. He's now working in my laboratory. And then these guys, Agota did a lot of experiments on the SVM-KTX, and Umer did a lot of experiments on the CN28 that unfortunately I didn't have time to work on or is explained. And obviously, Lurival from the international collaborators, Christine Beaton and Ray Norton, we are working together on some projects. This is, by the way, Lurival getting the honorary doctorate degree from the University of Debrecen. This was done in Debrecen, in the University of Debrecen gown. With the, you didn't have a hat, did you? No, you had the gown. But it was a great honor to have Lurival there as the honorary doctor of the University of Debrecen. And then I also have some Hungarian collaborators, Katalin Kövér, who is doing NMR studies, and Gábor Tóth, who is doing peptide synthesis. And I would like to thank your attention with the supporters. And this is a very nice present, was delivered to Adam Bartók, who was working on the Anuroctoxin stuff. And on this PhD defense, he was served with a scorpion cake. So we made a cake, a scorpion out of the cake. Okay, so thank you very much for the attention.
Thank you, George, for such a nice presentation. Really very interesting. There are time for questions if anybody wants to ask. Yeah, good. A ver.
Thank you, Gyorgy, for your talk. I have a few questions which will lead all to the same main question. And this is regarding the therapy. You know, which is what you want at the end.
Yes, this is what we are aiming for.
Okay. The first one is about the distribution kinetics of the peptides, of the toxins, once you inject them in the individual. This is the first one. The second is, so you have a distribution that will lead to only local distribution, to the blood distribution, or will get to the tissues where you have the affection. For example, an autoimmune disease of any kind, okay? This is the first one. The second is, do you have any idea of the kinetics of elimination of the peptide once you have it in the individual? Because it's a very small molecule and it's supposed to be eliminated through the kidneys and urine. So one would expect that the half-life inside the body will be short.
Yes.
The third one will be about the destiny of those Kv1.3 ion channels in the effector memory cells, T cells. Once they interact with the blocker, do they get internalized? Do they get metabolized? So the overall amount of number of Kv1.3 in those effector memory T cells gets lowered. So the effect is protracted in time. So you don't have to give the patient very frequent injections of the toxin.
Yeah.
Because you will have those, or will they die, or what will happen to them? Those three questions point to the main questions. How often would you need to give the patient that treatment in order to limit the consequences of the autoimmune disease? Thank you very much.
So it's thanks for the questions, and I'm really glad that you were not the reviewer of my grant, because then we wouldn't get the grant for sure. Now these questions are something that are bugging us for a long period of time. So distribution, local injection. Very interestingly, if you are, nobody knows why, but upon the injection of these toxins, if you do subcutaneous injection, for example, there is a depot for unknown reason. So nobody knows what will affect whether the toxin is going to be complex with some of the glycosaminoglycans or some of the connective tissue building blocks. But once you are injecting it, and there are many instances or descriptions of that. So the local injection will create a depot. From that depot is going to be a gradual absorption of the peptide, and that gradual absorption for the peptide will allow you to, the re-injections are not very frequently needed. I mean, once or twice a week, if you inject even once a week, injection of the peptide can have very significant therapeutic effects due to the formation of these small deposits or depots. If it was given, you're completely right. If it was given intravenously, you would have a half-life around 20 minutes. So the toxin is so small that it's going to be filtrated through the kidney and it's going to be eliminated through the urine. That is where the depot is going to be helping in the tissue during the local injection. And then for the third question, what happens to the molecules? So it's Kv1.3 when it's inhibited. The reason why we're developing these Cy5 conjugate toxins is that the guy, Ray Norton, is very much interested in the kinetics of Kv1.3 trafficking. What happens when you are conjugating the peptide with the toxin, and the toxin inhibited channels, what happens to them? Do they get internalized? Do they get internalized in a higher rate? Thereby, you are reducing the Kv1.3 expression of those cells. This is what the next step is going to be once we have already created this very high affinity Cy5 conjugate Kv1.3 inhibitor. So I cannot answer that question, but we are doing now confocal microscopy and trying to figure out how the Kv1.3 is going to be internalized. So these are going to be the issues that are going to be affected very dramatically the outcome of the Kv1.3 based therapy. There is one very interesting paper. It's in bioarchives. I don't know whether you had a chance to see it, and this was also the very interesting information to Professor Posani, that in Christine Beaton's lab, they developed small an expression system from some kind of lactobacillus. They incorporated the SHK gene into that bacillus. So they were given orally to the animals, and then the animals, the bacteria ingested, they are going to be helping to bypass the stomach. So they end up in the intestines, and in the intestine, the bacteria start to produce the toxin, and this is high enough concentration that the toxin can be absorbed. And they can get very good plasma levels of the toxin just by intaking the bacteria rather than injecting parenterally or any other means. So this is going to be probably a breakthrough in the Kv1.3. I mean, this is something which is better than we had before. This one is making the disulfide bonds. I mean, I'm going to be sharing the paper with you. This is a very nice bioarchive paper, which is already submitted to some top journal. They make these bacteria. I know that bacteria are not very good to make the disulfide bridges. This bacteria is going to be making in a functional form. So what they did is the bacteria was injected, not injected, intaken by the animals, and then the SHK absorbed through the guts, still don't know how, but they could get plasma level and they could do a functional assay, not only biochemical. What they did is they isolated the plasma from the supernatant. They did to electrophysiology, and they showed that the SHK is going to be inhibiting the Kv1.3 current. So this gives you the idea that most likely not 100% of the toxins are going to be properly folded, but a significant part of the toxins is going to be very properly folded. So this is what we think that is going to be very important for therapy. But you're right, all these questions are extremely important to target. Pharmacokinetics, either by the formation of the depot or the absorption from the gastrointestinal tract, they are both very good. You know, the only drug that is used currently in the therapy is Prialt, which is you have to put it into the intrathecally. So that's very problematic. So it didn't go to a very wide population because intrathecal administration of the peptide is extremely difficult. Thank you for the question, but this is very, very important.
You have another question.
Yes.
Thank you very much for your great talk. I have a question about physiological function of Kv1.3, because usually potassium channel plays inhibitory role for cell excitation, but in the case of T cell, potassium channel is involved in activation.
Yes.
So I'm not familiar to.
I didn't want. I understand. So what happens is that when let me see if I have a better figure. I probably So this is the. Can you turn the lights off a little bit so that we can use this? Yeah. So this is how we imagine a T cell. So the T cell has a lot of ion channels in the membrane, especially KCA3.1 and Kv1.3. What happens is that these ion channels, when they are operating, they will keep the membrane potential negative. Now, this negative membrane potential, which is around minus 50 millivolt, is needed for calcium entry into the cell. So this is the indirect. Role of these potassium channels is that they maintain this. We call it the so-called the permissive membrane potential, which will allow calcium to get in through the calcium release activated crack channels. So these crack channels will be the function of these calcium entry pathways will depend on the membrane potential. If it's not negative, if it's depolarized, depolarization will inhibit the calcium entry through the crack channels.
This point is not. I'm not very well convinced because the driving force of calcium is sufficient.
Yes.
Minus 50.
Now, what you are forgetting is that when calcium enters into the cells, it's going to be making these calcium-rich microdomains under the membrane. So calcium gradient in general is very large, but locally at the membrane is very, very small because calcium is getting in. It's going to be making this calcium-rich microdomains under the membrane. Therefore, the driving force for calcium will decrease due to the equilibration of the calcium concentrations. The only thing what remains is the negative membrane potential that must drive the calcium ions into the cell. So this is shown very clearly. There is a German guy, I don't know his name, top of my head, who who did very high resolution dynamic imaging of the calcium signaling, and he showed that upon the opening of the crack channels, you will have almost equilibration of the calcium very, very quickly. The concentration gradient.
Okay, I should learn more.
No, I would be very happy to talk to you about that. But this is the whole principle. You're right. This is against reality. Reality would dictate that calcium has such a large gradient that it doesn't matter how much the membrane potential is. Indirectly, it will matter because the calcium distribution is going to be very, very quickly equilibrating the calcium gradients. Therefore, you need the extra membrane potential. And this study was first done by a Hungarian guy called Balázs Sarkadi. He published this, that depolarization inhibits calcium signaling. He published this in the 1980s, but everybody forgot about it. Everybody thought that it's bogus because the same reasons. And then it turned out later in the 2000s that it is indeed the case. And we also show it ourselves very systematically that there is a inhibition of the calcium influx.
Really unusual.
Unusual, very unusual, very unusual. This is for neurobiologists, they always get crazy about it. So that's impossible.
Thank you very much.
Is there another question? Another? No more question? Well, so we thank you very much for this nice presentation.
Thank you very much for the opportunity. Thank you very much.